I will just start in 5 minutes. We'll wait to see if anyone else joins. Okay, so I'm sharing my screen. Are you able to see this? Hello? Hello, are you able to hear me? Can you just unmute yourself for once and let me know that you can hear me?
Yes, ma'am.
Okay. So I'll just start. Please admit anyone else if they want to join. So just click on the admit button. I'll start. So this slide is visible?
Yes, ma'am.
Okay. So basically what will happen in this session is that for the first 1 hour to 1.5 hours, I will talk about the first week lectures, a summary of the lectures that sir has taught, and you can clear all your doubts. And for the next 45 minutes or so, it will be like we will solve some practice problems. So this is, I'll start now. So firstly, this is the summary of week 1, lecture 1. That lecture, it focused mainly on basic things required to understand this course and on the history of biological inventions. Just stop me anytime you have any doubts, or if you had any doubts about the class also, you can ask about them here. Okay, so structural biology is the study of how biological molecules are built. So from the specific molecular function of a protein, it comes from the 3D structure of the protein. So if we know the structure, we can find out about the function. And if we know the sequence of the protein, meaning the different amino acids, we can find out about the structure. So structure, sequence, and function are interconnected. Now, based on the function, evolution of a protein is also dependent on the function. So basically what happens in evolution is that those organisms that are more likely to survive and reproduce, those are the organisms that continue to pass their genes to the next generation. For example, if a plant has more chlorophyll, it will be able to capture the energy of sunlight in photosynthesis, and it will be able to survive better, and so it will have more offspring. So depending on the function of the protein, the evolution changes the sequence so that the function can be better and better. So proteins are constantly evolving to optimize the function. Next, in lecture 1, sir had talked about the history of the instruments for visualization. So first, a glass sphere was used for magnification. Then at the end of the 16th century, the compound microscope was used, which had 9 times magnification. And in the late 17th century, this magnification was improved to 270 times. But these microscopes had a problem of chromatic aberration. So what is chromatic aberration? It is basically when the lens is failing to focus all the colors at the same point. So when the light containing 7 colors come, they get split up passing through the lens. And so the focal point is increased. And so instead of a sharp image, we will get a very blurry image. So how was this corrected? Basically, lens was taken with 2 different kinds of materials, crown glass and flint glass, which had different degrees of this resolving power. And so from this combination of lens, the colors which were splitting up this much, this was minimized so that the splitting is much lesser. So that is how chromatic aberration was corrected. And since there are 2 kinds of lens, this was called an achromatic doublet. And though the correction was not perfect, it managed to improve the resolving power. Then Sir had talked about Darwin's theory of natural selection. So Sir had not gone into the, please admit whoever is asking to be admitted. So yes, Sir had not talked about this in details, but I will help you understand that by an example that I've given here. So basically what Darwin's theory of natural selection had said is that because the resources are limited in nature, the organisms with the heritable traits that favor survival and reproduction will tend to leave more offspring than their peers, causing the traits to increase in frequency over generations. So what does this mean? So in a study was done in which there were many mice. Some of the mice were dark colored and some were light colored. Now what happened is that the dark colored mice, they could hide in the dark colored rocks and soil. So the birds of prey could not see them, and they ate mainly the light colored mice. So what happened in the next generation, the population of dark colored mice were many more because they were managing to reproduce. But since the light colored mice were being eaten up, they were dying out, and so their population was decreasing. So what happens is that only the dark colored mice were able to pass on their traits to the next generation. So basically, this is natural selection, and it causes the population to become adapted or well suited to their environments over time. So this natural selection, it depends on the environment, and it will always help in improving the ability of the organism to survive in that environment. That is why the light colored mice, they were not able to survive. So eventually they died out. Then in the first class, Sir had talked about laws of heredity by the father of genetics called Gregor Mendel. So the first law had talked about this law of segregation, which had stated that the hereditary units are paired in the parent and segregate during formation of gametes. So basically, this is the tall pea plant, which had the genes capital T, which stand for tall. And this is the dwarf plant with small t. So there are 2 gametes, and they are splitting up while forming the offspring. Then the law of independent assortment states that each pair of units is inherited independently of all other pairs. That means from this plant, the offspring of this plant can either get capital T or small t. And same for this plant. It is independent what comes from this plant and what comes from this plant. That is why the offspring can have 4 possible combinations. 2 capital T, 1 capital T, 1 small t. Similarly, 1 capital T, 1 small t, and both small t. So the law of dominance, it maintains that the trait units act as pairs. So in a paired with contrasting traits, the dominant one appears in the hybrid offspring, although the recessive one is also present. So basically, if you have 1 capital T and 1 small t, the capital T here is dominant, and so the plant will be tall. So this is what is meant by the law of dominance, is that if you have 2 different characteristics, then the dominant characteristic will predominate. So only if you have 2 small t, you can get a short plant. Then there were several experiments done to find out that it is DNA which is the genetic material. So the first experiment, called the Griffiths experiment, it talked about the pathogenicity of Streptococcus pneumonia. So the hypothesis was that there is some material in dead bacterial cells that can transform the living bacterial cells. So what Griffith had done, he had taken a living smooth strain or S strain of bacteria that can cause disease and injected it into a mouse, and the mouse had died. Then he had taken a living rough strain which cannot cause the disease, so it is non-virulent, and the mouse had lived because it is not a disease-causing strain. Then he had taken the disease-causing strain, but he had killed it by using heat. So then he had injected this heat-killed bacteria into the mouse, and the mouse had lived because the disease-causing bacteria was dead. But the most surprising observation was that he had taken the living rough strain and the dead smooth strain. That means the harmless bacteria was living, but the disease-causing bacteria was dead. But even then the mouse died. So there is something even in the dead bacteria that is getting transformed into the living bacteria and is causing the mouse to die. So this something was called the transforming principle. But whether this was RNA, DNA, protein, lipid, carbohydrate, that was not clear from Griffith's experiment. Does anyone have any questions so far?
No, ma'am.
Okay. So the next experiment was by Avery, MacLeod, and McCarty, where it was they had tried to find whether this transforming principle was protein or nucleic acid. So he had taken some of these heat-killed cells and he had removed the proteins and removed the lipids and sugars. Then in the first one, he had added the enzyme proteinase. So proteinase is breaking down all the proteins, so there is no protein. And then he had added the harmless bacteria, and he had seen that even though there is no protein, there is something that is something that is passing on to the new bacteria is still there. So protein is not the genetic material. He had done the same thing using RNase, which is an enzyme which breaks down RNA. And he had found that even then the transforming principle was present. Only when he had added DNase, which breaks down DNA, he saw that this transforming principle is not passing on to the R cells. So from this, he deduced that it is the DNA which is causing the genetic material to go to the offspring and not RNA or protein. So they established that the active genetic principle was DNA, and its transforming activity could be destroyed by DNase or deoxyribonuclease, which is an enzyme that is degrading DNA. The final experiment was by Hershey Chase. So what they had done is that they had taken this virus called a bacteriophage, and they had radio-labeled the proteins with 35 sulfur and the DNA with 32 phosphorus. And they had injected this radioactive elements into these bacteria of the bacteriophages into bacteria by adding by allowing them to infect the bacterial cell. Once this infection was done, they had done a process called blending, by which the viruses had been removed from the cell, but the DNA that was inside the viruses was injected inside. Then they had centrifugated and found that when the proteins had been labeled with the 35 sulfur, that was not detected inside the cell. So the protein is not passing into the cell. But the DNA that is labeled with 32 phosphorus, that is passing into the cell and causing the disease to the bacteria. So Hershey-Chase experiment confirmed that it is DNA, not the protein, that is the genetic material, because the radioactive protein was not found inside the cell, but the radioactive DNA was found inside the cell. Then this is the summary of the lecture 2. The lecture 2 basically talks about biological macromolecules. So there are 4 main classes of biological macromolecules. Firstly, it is the carbohydrates, and these are the immediate sources of energy for the living organisms. So monosaccharides are of many kinds. Here are 3 examples: glucose, fructose, and galactose. And the basic formula for them are CH2 whole N, because it is carbohydrate, carbon, and H2O, which is hydrate. And these are the simplest forms of the carbohydrates, and so these are their building blocks. Because they have multiple hydroxyl groups, they can form a dehydration reaction between 2 hydroxyl groups and join them through a glycosidic bond. And so this is a disaccharide called a sucrose, which is made up of glucose and fructose. This is lactose, which is made up of 2 monosaccharides, galactose and glucose. And these are classified on the location of their carbonyl groups and on the number of carbons. And this is a polysaccharide where multiple innumerable numbers of monosaccharides are attached. This is glycogen, which is made up of numerous numbers of glucose units. The second biological macromolecule is fat. So fat is mainly made up of glycerol, which has 3 OH groups, and fatty acids, which is a long aliphatic chain and a carboxylic acid group at the end. So these combine through an esterification reaction to form an ester bond. So if you have the glycerol and 3 fatty acids connected through an ester bond, this is called a triglyceride. And this fatty acid chain, as sir had discussed, it can be saturated, meaning it is all single bonds, or it can be unsaturated. So there is a cis double bond between it. So whenever this double bond is there, there is a bend in the chain and the packing is not very good. So a straight chain can pack much better than this bent chain. Another type of fat is the phospholipid, where to the glycerol molecule, 2 fatty acids are attached by the esterification reaction, and to the third OH of the glycerol, a phosphate group is attached. And so since the phosphate group has a negative charge, this is hydrophilic. And since these are alkyl groups, they are hydrophobic tails. Another kind of fat is the steroid, and these are characterized by a carbon skeleton consisting of 4 fused rings. So you can see the 4 rings A, B, C, and D here. And this is cholesterol, which is one of the most common kinds of steroids in the human body. The third kind of biological macromolecule is the nucleic acid. So there are 2 main classes of nucleic acids called deoxyribonucleic acid, DNA, and ribonucleic acid, RNA. So nucleic acids are polymers of these nucleotides and called polynucleotides. You can see the nucleotides shown here. They are made up of an aromatic base, which is this moiety here, a pentose 5-membered ring sugar, and a phosphate group. So this aromatic nitrogenous base can be of 4 types. This is adenine, this is guanine, this is cytosine, and this is thymine. In RNA, instead of thymine, uracil is present. Another difference in RNA is if you look at the sugar in DNA at this position, there is an OH group. In this position in RNA, there is an OH group, but in DNA, it is an H, which is later replaced by the phosphate. So that is another difference between DNA and RNA. And these bonds that the nucleotides form are called through the phosphate, are called phosphodiester bonds. The fourth kind of biological macromolecule is the protein, and the basic unit of the protein is called the amino acid. So amino acids have an NH2 group, a COOH group, an H group, and an R group. And the R group in naturally occurring amino acids can be of 20 types. So all the 20 amino acids are shown here. And this R group can be very small, like just one hydrogen or one CH3, or it can be nucleophilic. That means it is an OH which has a lone pair, or an SH which also has a lone pair. This R group can also be hydrophobic, like different alkyl groups. Proline is the only cyclic amino acid. The R group can also be aromatic, like a phenyl ring, an indole ring. The R group can be acidic, like aspartic acid, which has a COOH, and a glutamic acid, which also has a COOH, but with another carbon between it. R group can also be an amide with a CONH2 group, or it can be basic, like histidine, lysine, or arginine. So all these amino acids have a 3-letter code, like glycine, it's GLY, and a 1-letter code, G. So this you have to memorize. All the 20 amino acids, the structure and this 3-letter and 1-letter code, you will have to remember eventually. Another thing that Sir had taught in the second lecture was the 3 secrets of covalent bond. Firstly is the chirality, which is the property of a molecule that is making the molecule and its mirror image non-superimposable. So if you look at this molecule and you pretend that this dotted line is a mirror and this is the mirror image, so you can see from here that the molecule and the mirror image are not superimposable, because here for this molecule the green atom is here, and for the molecule behind it the blue atom is here, and there is no way that you can superimpose these two. So this is also called configuration, when the spatial arrangements is such that to interconvert one into the other, you have to break these two bonds. So these two molecules have different configurations. And the third C is called conformation. So if you look at this molecule, this is basically, this representation is when you are looking from one side of the molecule. You can see that the two CH3 groups are parallel to each other. If you just rotate this single bond by 60 degrees, the two CH3 groups become at an angle of 60 degrees to each other. So you can easily interconvert between these two because there is a single bond between them and that can freely rotate. So conformation is such a spatial arrangement of atoms that can be obtained by the rotation about the single bond. Next, we come to lecture 3. Lecture 3 basically talks about decoding the biological macromolecules. So as you can see, 2 glucose molecules here, and a dehydration reaction is occurring, meaning a water molecule is removed to form this glycosidic bond. Now this glycosidic bond is a single bond, so free rotation can occur in this bond, like you saw in the previous slide, and it can give rise to a number of different conformations. So that is why structure prediction becomes very difficult in carbohydrates, because there can be so many different kinds of conformations because this is a single bond. Another difficulty in carbohydrates is that for different kinds of glycosidic bonds, different enzymes are required to cleave them. So the process becomes very expensive. Now, if you look at DNA, RNA, or proteins, their synthesis is template based. That means in the cell, we have the chromosome, and in the chromosome, we have the DNA. If we know the sequence of the DNA, we can find out the sequence of the RNA, And from the RNA, we can find out the sequence of amino acids that form the protein. So DNA, RNA, and protein are all related to each other, and it is template based. So once we decode the DNA, we can find out about RNA and protein. However, carbohydrate and lipid is not like that. And so it is very difficult to decode them. And another problem with carbohydrate or lipid is that, like you saw in glycogen, which is a polysaccharide, there can be 1 million molecules of glucose connected through glycosidic bonds. So there is no standard set for carbohydrates or lipids. Like there are 4 bases in DNA and 20 amino acids in proteins. So these carbohydrates and lipids are very individualistic. So even the same organism depends on what they are eating. The composition of carbohydrates and lipids will vary. But in all humans, the DNA set is similar. So that way also carbohydrates and lipids pose a lot of problems for decoding. So for now, we will focus on decoding proteins and DNA. So for decoding proteins, which is protein sequencing, a practical process of determining the amino acid sequence of the protein or peptide. In class, you had learned about Edman degradation. So this will, here you can see the actual chemical reaction for Edman degradation. So basically, this is the polypeptide, and phenyl isothiocyanate is added, which is this molecule. Now this molecule will attach to the N terminal of the polypeptide, and this is called a phenyl thiocarbamyl derivative. Then at acidic pH, what happens is that this sulfur will attack the carbonyl and the rest of the amino acid will be released, but the N-terminal amino acid will remain with this phenyl isothiocyanate. Enough, and it will form a 5-membered ring. So this is called a anilino-thiazolinol derivative of the amino acid. So the N-terminal amino acid is getting cleaved away to form this 5-membered ring, and the rest of the amino acid peptide remains as it is. Again, at low pH, this 5-membered ring rearranges to form a phenyl thiohydantoin derivative. So what this process has accomplished is that by adding phenyl isothiocyanate, the N-terminal amino acid has been cleaved, leaving the rest of the peptide unreacted. So you can selectively do mass spectrometry for the N-terminal amino acid and find out what is this amino acid. So now you know the N-terminal amino acid. You can keep repeating this process for the rest of the peptide by cleaving the next amino acid, finding out its mass, then cleaving the next amino acid, finding out the mass. And like that, you can find out the sequence of the entire peptide. Another way of protein sequencing is called mass spectrometry. And what is done in that is that the polypeptide is taken and it is fragmented in different ways by using proteinases. Please admit whoever is asking to be admitted. So in mass spectrometry, once you fragment the different peptides, there are fragments of different sizes. So there can be 4 amino acids, 5 amino acids, and then you take the mass. So in the mass, you will get different peaks for the different size fragments. And so for the smallest, the peak which is at the lowest mass will be only for 1 amino acid. The one that is for 2, for the next peak, it will be for 2 amino acids. Like that, you can assign the peaks, which peak belongs to which fragment. And from that, you can find out the sequence of the polypeptide. So these are the 2 methods, Edman degradation and mass spectrometry for protein sequencing. Now to learn about base pairs. So a few slides ago, you saw that DNA is a polymer of the 4 nucleotides, A, C, G, and T, which are joined through a backbone of alternating phosphate and deoxyribose sugar residues. These nitrogen-containing bases occur in complementary pairs and form hydrogen bonds between them, shown by these dotted lines. Adenine always bonds with thymine through 2 hydrogen bonds, and guanine with cytosine with 3 hydrogen bonds. So this structure, along with the molecule's chemical stability, makes DNA the ideal genetic material. And the bonding between these complementary bases, AT and GC, provides a mechanism for the replication of DNA and the transmission of genetic information. So next you learned about DNA replication. The first step of this was unzipping, in which an enzyme called helicase, which is shown here, is breaking the hydrogen bonds holding the complementary bases of DNA together. The next step is the complementary base pairing. So once the DNA is unfolded into 2 single strands, there are different nucleotides which are moving into position and coupling with the single strand. Like here, if there is an A, the T will come and couple with it. If there is a G, C will couple with it. And the final step is the formation of the sugar phosphate backbone. So once the nucleotides are joining, the sugars and phosphates are also binding to form a new backbone using the enzyme DNA polymerase. And another function of the DNA polymerase is that it will check that there are no mistakes in the DNA so that this whole process is error-free. Next comes the central dogma of molecular biology. Basically, the central dogma means that DNA can form other DNA through replication. From DNA, RNA can form through transcription, and from RNA, the protein can form through translation. And this is how life is interconnected, and it's called the central dogma. The polymerase chain reaction, or PCR, is a method by which large quantities of DNA are amplified from small quantities. So first, what happens is that the double-stranded DNA is denatured, that is, the 2 strands are separated by heating this DNA to temperatures about 95 degrees Celsius. So each strand is a template on which the new strand of DNA will form. The next step is called annealing. The temperature is reduced from 95 degrees Celsius to 55 degrees Celsius so that the primers can anneal to the template. So a primer is basically a small oligonucleotide sequence, and before any of the nucleotides join, the primer needs to add to the DNA, because DNA polymerase can add a nucleotide only when there is a pre-existing 3' OH group. That is why initially a primer is always needed for this DNA replication or polymerase chain reaction. The third step is called extension, where the temperature is again raised from 55 degrees Celsius to 72 degrees Celsius, and the DNA polymerase keeps adding these nucleotides one by one to form the rest of the DNA. So in one cycle, from one DNA, you can get two DNAs. In the second cycle, from 2 to 4, from 4 to 8, and so on, in the nth cycle, you can form 2 to the power n copies. So polymerase chain reaction is forming a huge number of copies from a very small amount of DNA so that other experimental techniques can be carried out. And a method of this DNA sequencing called Sanger sequencing was also discussed, which is the process of determining the nucleic acid sequence. So this DNA that we want, the DNA of interest, is used as a template for a special type of PCR called the chain termination PCR. In chain termination PCR, you use a low ratio of chain terminating dideoxyribonucleotides with the normal deoxyribonucleotides. So basically, this is the normal deoxyribonucleotide, which is a monomer of the DNA. And here, the OH group at the 3' is removed to form a dideoxyribonucleotide. So now, whenever this dideoxyribonucleotide is present, the DNA extension is going to stop because there is no OH group, so the phosphodiester bond is not possible. So when DNA polymerase will incorporate will one by one bring the nucleotides to pair with this template DNA. So whenever it reaches a dideoxyribonucleotide, the extension will stop. So the result of this chain termination reaction is millions to billions of oligonucleotide copies of the DNA of interest, which is terminated at different lengths. So you can get 3 nucleotides, 5 nucleotides, 10 nucleotides, different lengths of this DNA whenever they are getting terminated by this dideoxyribonucleotide. In the second step, these different oligonucleotides are separated by size via gel electrophoresis. So they are passed through a gel, and in the gel, the large nucleotides will move slowly, so they will remain on top, while the small nucleotides will move fast and they will be separated. So after this size separation, the last step involves reading the gel to find the sequence of the DNA. In lecture 4, Sir taught an improved sequencing technique called next generation sequencing, or NGS. So what is done in this is there are 4 steps. Firstly, the nucleic acid extraction and isolation. So the extraction method used is optimized to yield the maximum amount and highest purity of nucleic acid. After the second step is library preparation. This is done by fragmenting the samples to get a pool of appropriately sized targets and then adding specialized adapter DNA at both ends. So these colored fragments are called adapter DNA, and they are artificial pieces of DNA introduced prior to sequencing to ensure that the DNA fragment being sequenced attached to the sequencing flow cell. So these adapter DNA are not DNA that you want. It is basically attached to ensure that these oligonucleotides are sticking to the sequencer, because only when it sticks to the sequencer can it find out the nucleotide sequence. So these prepared ready to sequence samples are called libraries. The third step is sequencing. So after amplifying the DNA fragments to be sequenced, the library is loaded onto the sequencer, which reads or detects the nucleotides one by one. So for the different fragments, the sequencer can find out what are the nucleotides in each fragment. And step 4 is called data analysis using bioinformatics. So what it does, it takes these nucleotide sequences and it finds that which are the common areas. So basically you can see this area is AATATC. So that is part of the DNA. Then to confirm that it is correct, you can see that both fragments have this 5Cs portion, which is next comes in the part of the DNA. So like this, from the different fragments, it is reconstructed and the entire polynucleotide sequence is found. So we have learned about Sanger sequencing and next generation sequencing. But what is the difference between them? So basically, in next generation sequencing, millions of short DNA are read parallelly, while Sanger can only read a small portion of the DNA at a time. In next generation sequencing, you are fragmenting the whole DNA and reading it at the same time. The speed of the sequencing process is also much higher than Sanger sequencing in next generation sequencing. Basically, with improved software, the speed has increased a lot in the next generation sequencing. The cost is also low. And in Sanger, an additional step was required, which is the gel electrophoresis. But in next generation sequencing, it is directly read because these fluorescent labeled nucleotides are incorporated, and through laser, it can detect which nucleotides are coming in which sequence. So there is no need for a gel electrophoresis, which is an additional step in Sanger sequencing, and so it takes extra time and money. In lecture 4, Sir also talked about the Human Genome Project, or HGP. So the Human Genome Project was an international research effort to determine the sequence of the human genome and identify the genes that it contains. There were many aims of this Human Genome Project. Firstly, to sequence the whole genome with 3 billion base pairs. Secondly, creating a physical map of the human genome. Then, to identify all the disease-causing genes. The fourth was to understand the function of these genes and to make the information available to all researchers. And finally, to develop tools for processing and analyzing data. However, even after the Human Genome Project, there were many problems. Firstly, after genome sequencing, we have millions and billions of the gene sequence. So from the gene sequence, we can know the sequence of many proteins. But we still do not know the 3D structure of these proteins. So that is a problem that is present even after the Human Genome Project. That is, we need to know the 3D structure of the proteins to find out about their function. Then you had learned about two different databases. A database theoretically refers to a set of related data and the way it helps the data to be organized. So for protein 3D structures, the first database established in 1971 was called Protein Data Bank, or PDB, where from all areas of the world, proteins of different structures were being deposited in that protein data bank. The PDB ID is generally 4 numbers and letters, with the first always being a number. And the higher this number, the more recent the structure. So between the 3 PDB IDs given, this 4 is the most recent, then is 2 GDN, and the oldest is this 1 JTG. Now, one protein can have multiple PDB IDs because one protein can have many different conformations, so in different environments. So according to that, one protein can have as many PDB IDs as the number of conformations. The protein database for the protein sequences was first established in 2002, and it was called UniProt, or Universal Protein Resource. The UniProt ID is generally a combination of 6 letters and numbers, but can sometimes be a combination of 10 letters and numbers. Now, the UniProt ID is unique for a protein because a protein can have many different conformations, but it can only have one sequence. So as you can see here, these are different kinds of proteins and they all have different conformations. This has 3 helices here. Here the conformation is changed, the helices become shorter, but the sequence is always the same. In the final lecture of the introductory class, Sir talked about the post-genomic era, which started after completion of the Human Genome Project and push the journey from sequence understanding to functional understanding. So from the DNA sequence, by transcription and translation, you can come to the amino acid sequence. And from the amino acid sequence, by folding, you can get the 3D structure of the protein. But you need to know the 3D structure to find the function. So the challenges in the post-genomic era was basically that what is the importance of this folded protein structure? How from the structure you can predict the function? And why it has taken so much time and money to solve this structure? And and you can see from the time graph that Sir had shown that initially the structures were known much more than the sequence. But now what has happened is sequence is rapidly being known, but the structure is still lagging behind. So this is a challenge in the post-genomic era that we need to find better techniques to find the 3D structure of the protein. He had also talked about 3 methods of determining the 3D structure of proteins that are present. Firstly, the X-ray crystallography. So what is done here is first the protein is crystallized. And this is the most difficult part of the process because a perfect crystal structure is needed so that the position of the electrons can be mapped accurately. The second step is production of a diffraction pattern. So a diffraction pattern is formed on the screen by the X-rays, which have been absorbed by the atoms in the crystal, and this leave behind dark diffraction spots. So a diffraction pattern is shown here, and the density of these spots varies with the amount of interference between the diffracted electrons at each point. So from the diffraction pattern, you can find the electron density in the crystal. Then this diffraction pattern is analyzed to form an electron diffraction map. Electron density map. So basically where the spots are more, that is an electron dense region. So probably it can be something like a phenyl ring, which is very electron dense because you know the phenyl ring has pi electrons. So like that, the diffraction pattern is interpreted to form a map of the electron density. And then from this map, there is determination of the protein structure. So using mathematical calculations called Fourier transformation, the data is transformed into the 3D representation of the atomic or molecular structure. So from this electron density map, you can predict the atomic model, like how many helices there are or how many beta sheets. And again, from the atomic model, you can get a better diffraction pattern and repeat this process many times to get a refined atomic model. The second method of determining the protein 3D structure is nuclear magnetic resonance, or NMR. So in this, what has happened is that protein is grown in a separate media which has these labeled nuclei, 1H, 13C, 15N, or 31P. So when the protein is grown in the presence of these labeled nuclei, then it can be detected by NMR. So NMR can differentiate between atoms in different chemical environments in an external magnetic field and identify the distances of nearby atoms. So this labeled protein is run through an NMR machine, and 1D, 2D, and 3D NMR is found. So what happens is NMR, the distance between two atoms can be calculated. So whenever two atoms are interacting, you will find spots like this in the NMR. And from these spots, the intensity of these spots, you can find the distance between two atoms. And like that, you can reconstruct the structure of the protein. But NMR has many problems. So basically, when a molecule is tumbling, that is rotating and moving very fast, then the resolution of the NMR is very good. But when the size of the molecule is large, like most proteins, the molecular size is very large. So the protein moves very slowly. So the resolution decreases. This resolution can be improved a little bit by taking higher magnetic strength of the instrument. But even then, there is a restriction of the molecular weight. So the problems with NMR are shown here. Firstly, the protein must be soluble at a high concentration and stable without aggregation. And as I said, there is a restriction in the molecular weight because with increase in the molecular weight, the tumbling is slowing and the resolution of the NMR spectra is decreasing. So a third technique was found, which is cryo-electron microscopy, or cryo-EM, where there is no such restriction of the molecular weight. So cryo-EM has 5 steps. Firstly, you need to purify your molecule. So for high quality results, the sample should be purified and structurally intact. The second step is called plunge freeze, where the specimen is freezed so it doesn't freeze dry in the microscope's vacuum. Because suddenly if you put the specimen in the microscope, it will freeze instantaneously and any water in the protein will be eliminated. And these water molecules are essential to maintain the protein structure. So you need to freeze the specimen slowly by plunge freezing. And you apply a small amount of this frozen specimen to the grid of the transmission electron microscope and remove the excess liquid with blotting paper. Then you plunge this grid into liquid ethane to wet the specimen and freeze it. After you have done that, you transfer the grid having the frozen specimen to a transmission electron microscope, or TEM. So you transfer the frozen specimen to a specialized TEM holder to maintain liquid nitrogen temperature. To avoid any contamination, people use a cryo workstation to load the sample into the holder and a cryo shield to transfer it to the microscope. Then data is collected, so you can get a 2D projection. And then you go from particle alignment and averaging, you can get the view of the protein from different angles. So basically, say one portion of the protein looks like a square, another from the top view, it looks like a circle. So you get different 2D views of the protein. And by this imaging, so low dose imaging procedures are used to navigate and focus before taking these images. This enables us to visualize the water molecules, ions, and ligand structures in the 3D particle reconstruction. So from 2D, you get a 3D idea. And finally, analysis and reconstruction is done. This is done with a software called Gattan's microscopy suite, and you can analyze and export the data in various formats. It can be imported into a wide range of third-party software packages for 3D construction and visualization. So you have finally reached the 3D model of the protein. So this is the summary of the week 1 lectures of SIR. So if anyone has any doubts about the week 1 lectures, you can ask them now. Any doubts? Hello, am I audible?
Ma'am, actually, am I audible now?
Yes, you're audible.
Ma'am, actually, you have told about Edman degradation.
Yes.
Ma'am, my question is like how we will be able to know like when it will be terminated? Like if we get all the amino acids one by one, then how we will be able to know then that what is the actual length of the amino acid? Because in that way we can get a very long amino acid also and very short amino acid also, like a chain of very long amino acid and chain of very short amino acids also. Then how we able to know about the when to terminate it and what is
So,
the
okay.
actual
So you have to
what is the
constantly do mass spectrometry. So firstly, you terminate only one amino acid from the N terminal, and there is HPLC. So by HPLC is a method by which you can separate out the terminated amino acid only. So only 1 second. So you carry out this reaction one time and you will get only the N-terminal amino acid separated out by HPLC. So then you give it for mass spectrometry and you will get the mass of this. And from the mass, you can find out that which amino acid is there in the N-terminal, because the different amino acids will have different mass. So you know the N-terminal amino acid. Then you carry out the reaction another time and you will get the amino acid that is there after the N-terminal. So like this, by carrying each time you carry out the reaction, you have to separate this amino acid and find out its mass. So like that, one by one, you will finally be left with only 2 amino acids. And the final time you carry out the reaction, you can get the mass of the last 2 amino acids. So like that, you have to do it. Does that make
okay, ma'am.
sense?
Yes, ma'am. Yes, thank you.
Sure.
Ma'am,
Anyone?
ma'am, another yes, ma'am.
Mhm.
Another question. In NMR, you have told about the aggregated molecules. Like the problem is we will get the aggregate molecules.
Mhm.
But ma'am, my question is we are counting the nucleus only. Then, ma'am, the aggregated nucleus, the in aggregated molecules, there should be the aggregated nucleus also. So
Mhm.
our result will be, I think, amplified. Then it will be helpful for us to determine the structure. Like in lecture it was like when the X-ray crystallography we have seen, ma'am, actually
Mhm.
the amplified result, the crystals when they are amplified and they are giving better results in spite of a single molecule. So my question is, ma'am,
Okay,
when
so
why it will be problem?
yes, so in X-ray crystallography, it is in solid phase. So if there is an aggregation in a crystal, there is no problem because it is in solid phase. And so like the conformation will not change. In crystal, you know that a crystal is made up of repeating monomers, and all the monomers have the same structure. So in a crystal, if you have aggregation, then there will be like all the aggregated molecules will have the same conformation. So in X-ray crystallography, if there is aggregation, uh, it will give a better signal. But NMR is done in solution phase. So in solution phase, if there is aggregation, often the conformation can change. Like suppose if the protein is there as a monomer and it has a helical conformation, but two helices can aggregate and form a super secondary helical structure. So you will learn about that later. But the point is that in solution, if there is aggregation, the overall three-dimensional structure can change. And so it will give an NMR result which will match that of the aggregated structure, but you want it for the non-aggregated protein structure. So that is the problem with aggregation in NMR, because it is in solution state.
Yes, ma'am. Thank you, ma'am. Got it. Thank you.
Okay. Any other questions? Anyone?
Ma'am, I have one small doubt also, ma'am. Like
Okay.
when you are you told about the electron density we got, then the electron density of the diffract, like after getting the diffracted before getting the diffracted pattern, the X-ray uses the they attack the existing electrons and we get the diffraction pattern. But ma'am, my question is which atoms will they will attack the atoms of the crystal or the atoms of the proteins that is crystallized?
So the atoms of the proteins are the atoms of the crystals. I am not understanding your question, because the protein is
Because
your
ma'am,
first. Okay. What are you saying?
because ma'am, when we do the X-ray crystallography, we add some precipitants also, ma'am. And also we do the things in molten globule state, as
Okay.
told. My question is, are they the diffracted pattern we get? They are from the precipitants and the protein only, or only the protein structures we get the
Okay.
electron density?
So the machine that is giving you the diffraction pattern, it has a setting which will automatically cancel out the precipitant and the molten globule structure. It will only give you the diffraction pattern for the crystal. So basically what you have to do, you have to take two diffraction patterns. One will be without the crystal, which will be the baseline, and one will be with the crystal. And then you have to subtract the baseline from the diffraction pattern of the crystal, so that all these excess of the precipitates and molten globule will be subtracted out, and your final diffraction pattern will only be that of the protein crystal.
Okay, got it. Thank you.
Other questions? So first we'll clarify all the doubts, and then there are some sample problems for solving. So if you have any doubts, you can say that now, otherwise we will move on. We have a lot of time, 1 more hour is there. So just any doubts in any of the lectures also, like not only in this summary, but in any of sir's lectures of week 1, if you have any doubts, you can discuss them now. So will I move on to the problem solving part?
Yes, ma'am.
Okay. So these are some problems from previous assignments. So this is the first problem. Can anyone tell me the answer?
Well, this is B.
Actually, sorry.
This is B, option B.
Okay, so actually the correct answer is C, and I will discuss that now. So why the correct answer is C? So basically light microscope, this is the mechanism of a light microscope. What happens is that light is produced from an internal or external light source, and it passes through the iris diaphragm, which is a hole which controls the amount of light reaching the specimen. The light passes through the condenser, which focuses the light on the specimen, and the slide is held at 90 degrees to the path of the light so that it can get the maximum light. Then this objective lens magnifies the image of the specimen before the light passes through the microscope. And finally, the light reaches the eye and it sends signal to the brain so that you can see the image. So a very small specimen is magnified into a large image. So this is the principle of the light microscope, but since your eye is detecting it, the light must be a visible light. So if you look at this, the visible range is only between 700 to 400 nanometer. But the structure of proteins and other macromolecules is between 10 to the power minus 8 to 10 to the power minus 7 meters. So clearly, your visible spectra, the wavelengths are much larger. And all these biological molecules, they will fall very much between these wavelengths. So the wavelength of light cannot detect it. So coming back to the question, it is both A and B. So it is the range of light microscope is less than 100 nanomolar. And the biological macromolecules are in the range 10 to 0.1 nanomolar. So the correct answer is actually both of the above, A and B. So I'll go to the next question, or is there any doubt with this question? Okay, I'll go
No
to the
doubt.
next. Okay, I'll go to the next question. So this one.
Option D.
Yeah, it's nucleic acid, option D. So this is very clear, like I talked about it a few slides ago. So DNA is a nucleic acid and it is a polynucleotide made up of different nucleotides. So this one.
Proteins.
Yes. So the 4 biological macromolecules: proteins, carbohydrates, fats, and nucleic acids. So the correct answer is protein. DNA replication involves. So which one will this be?
All of the above.
Yes. So these are the 3 main steps as I discussed. So unzipping of DNA, then complementary base pairing, and then formation of the new sugar phosphate backbone. This is a bit difficult actually, but so I will discuss this answer properly. But does anyone know this? I think I'll discuss this answer. So basically, this correct answer is A, and I'll discuss this. So there is something called triplet coding, which is basically in DNA, 3 nucleotides code for 1 amino acids. So there are 20 amino acids, but only 4 bases: adenine, thymine, guanine, and cytosine. So you know, from DNA we get RNA, and from RNA we get protein. So the protein is already coded in the DNA. And the way this is coded is that three bases are required to code for each of the 20 amino acids. And this group of three bases is called a codon. So from the first three, we get one amino acid, from the next, another amino acid, and like that, we get a polypeptide chain. So if you look at this chart here, it is that which bases are coding for which amino acids. One thing you will notice is that genetic code is degenerate. That means that for the same amino acid, like serine was in the question. So serine has 6 possible combinations of nucleotides that can code for it. So why was this degeneracy necessary? So basically, if only there was like 1:1 correlation, meaning 3 nucleotides, 1 amino acid, if the correlation was like that, then there would be a big problem, because there are total 64 possible base triplets. So only 20 would code for the amino acids and the other 44 would be useless. So if 44 base triplets are useless, you cannot get the protein from the DNA so easily. So there has to be more than one triplet coding for the same amino acid. Otherwise, while decoding from the DNA to the protein, it will get truncated. So for most amino acid, there is more than one triplet that is coding for it. And for serine, if you saw the options, I'll go back to that. AGT is coding for serine. So this is actually a very difficult question to ask, because I don't know how it's possible to remember all this, but this was actually one of the previous year's questions. So I think maybe for the important amino acids, meaning like the amino acids where there are many codes, like serine has many codes, maybe those you can try to remember. So that is the explanation for this question, that which nucleotide is coding for serine. Okay, this is question 6, that what is the key feature of the Sanger sequencing method? Just try this.
Option B.
Yes, it is a dideoxy or chain termination method. I'll go to question 7. What are the experimental methods to determine the 3-dimensional structure of a protein?
X-ray crystallography. X-ray crystallography.
Yes. So there are 3 methods: X-ray crystallography, cryo-EM, and NMR. But for this, only the X-ray crystallography is the option is there. So that is the answer. Then question 8 is asking that which was the first protein to have its structure determined by X-ray crystallography?
Myoglobin.
Myoglobin.
Yes, it's myoglobin. And just to talk about the history a little bit, is that this was the original myoglobin structure that Kendrew had developed. And the structural study of myoglobin started in parallel to that of hemoglobin. And Max Perutz was also working on hemoglobin at the same time that Kendrew was working on myoglobin. But since the size of myoglobin was much smaller, like 1/4 the size of hemoglobin, the myoglobin structure was discovered first. And then they found, so from this structure, if you want to see an all atom structure, this was the structure of myoglobin with the all atom representation. And this is called a ribbon representation, where it shows that the myoglobin structure is mainly made up of helices, which are right-handed. And the molecule is basically a helix, then a loop, which is an irregular section, then another helix, another loop. And there is a heme moiety here. You will learn more about the heme moiety later. So basically an iron atom is there and a porphyrin ring. So myoglobin was one of the first protein structures to be placed in PDB when it was begun in 1971. And the PDB code was 1MBN. And since then, many structures have been deposited in the PDB for myoglobin itself. So like I said, one protein can have many different structures, like say in different solvents or other environments, myoglobin can have other structures. So for and also for different species of animals, the structure of myoglobin will be different. Like if an animal is very fast moving, like the cheetah, then the myoglobin amount will be more, and the structure will be such that it can carry more oxygen. And the myoglobin structure is also different in different species. So there are 400 different structures for myoglobin itself in the PDB. This is another question, which is saying that lactose is an example of.
Disaccharide.
Yeah. Correct answer is A, and it is actually, you have to remember that it is a, like the question can also come like this, that it is made up of which two monosaccharides. So that also you have to remember, like it is made up of galactose and glucose. So this bond is called a beta 1,4 glycosidic bond, because for galactose it is from the carbon labeled 1, and for glucose it is from the carbon labeled 4, in which there is a dehydration reaction and it forms this beta 1,4 glycosidic bond to form the disaccharide lactose. And another is that which bond is formed in the polymer formation of nucleotides in DNA.
Phosphodiester bond.
Yeah, that's a phosphodiester bond. And so like you learned that this 3' OH and the OH of the phosphate group, it does a condensation reaction. So the water is removed, and this is called a phosphodiester linkage. So I don't have any more slides. So if you have some more doubts, you can clear them, or I can show you that how you can visualize a protein structure in PDB. So I can do that. So just let me know.
No doubt. Actually.
Okay. If you want, you can stay for this section. I'll show you how to use a software called Discovery Studio. Does anyone have Discovery Studio?
No, ma'am. Actually, I don't have. I have only PyMol, ma'am.
Ah, okay. So anyway, I'll show you. I think Discovery Studio is more user-friendly than PyMol, and it's a free software. So because we still have around 45 more minutes, I can show that, like how to visualize that to you.
Yes, ma'am. Please show.
Okay. I'll download. What did we learn about the structure of, let's see myoglobin itself. So the code was 1MBN. Have you downloaded files from the protein data bank, anyone? Anyway, you have to go to download files and this PDB format. So you can see the structure of myoglobin here. And Discovery Studio, if you go here, you can see the amino acid sequence. So I think now the protein data bank has evolved, so that previously it was that protein data bank only had the structure and UniProt had the sequence, but now protein data bank has the sequence also. So Discovery Studio allows this, that so you can, sorry?
Can you repeat please once? Actually, network is with please.
Okay. So did you see like how this structure was gotten from the protein data bank?
No, ma'am. I need want
Oh,
to see.
okay. I'll go back. So in Google, you can just type 1MBN, which is the, or whatever is your PDB code. This is the PDB code for myoglobin. And you will directly get this. This is in the protein data bank. You can see it's written PDB here, and it will give you all kinds of data. So what is the classification of the organism, when it was deposited? See, this was deposited by Kendrew, and then the all the total structural weight and everything is also given. So you have to go to download files and download it in PDB format. So because I think PyMol is also allows the visualization of the PDB format. So once you download it, if you have PyMol, you can open it in PyMol. I have opened it in Discovery Studio. So Discovery Studio, you can enlarge this portion, and you can see that there are two portions of the molecule. One is this helical portion, which is the protein, and you can switch on and off the different regions. So if I switch off the protein, this is the inorganic part of the molecule, which is called a porphyrin ring. So this is basically the portion that is responsible for carrying the oxygen. And you can rotate the molecule and see it is made up of, like Kendrew had said, helix, loop, another helix, loop. And like if you want to see the amino acid sequence, there is this plus symbol here. You just enlarge it. It will give you the amino acid sequence. So now the protein data bank has advanced in such a way that previously it used to be that protein data bank has only the structure and UniProt data bank has the sequence. But now you can find the sequence from the protein data bank itself. So you can highlight particular amino acids, like I have highlighted this region. And this is called a ribbon representation. If you want to see the atoms which are there, you have to go to this display style, right click, then display style, and you can see this stick model or ball and stick model. So it will show the atoms. Is this visible?
Yes, ma'am, visible.
Okay. So you can actually check out different hydrogen bonding interactions, or like suppose this, if I want to add right now it is not showing the hydrogens. If I add a hydrogen by going to chemistry, hydrogen, add. So this is an NH2 group, NH2 group and a COO group. So maybe it can form a hydrogen bond. If we want to check that, we have to find the distance between these two atoms by selecting, selecting once, then going to structure, monitor distance. See, the distance it is giving the distance in angstrom, but this distance is too large for a hydrogen bond. Hydrogen bond should generally be between less than 4.5 angstrom. So this is not a hydrogen bond. So this protein is very well studied, but generally when people are developing peptidomimetics, which are, they want to develop a small structure for this myoglobin, they have to study these interactions very carefully. So in that case, they take the original protein and then they do this atom representation of all the amino acids. And then they try to find out which are the hydrogen bonds and all, so that they can replicate that. And you can build structures in Discovery Studio also. So there are different, if I want to build a molecule, Then I go to small molecules. Amino acids, the option you can see is here. So any amino acid you can add and form a chain of a polypeptide chain. And so you can build your own molecule also in Discovery Studio. So yeah, that is, and yeah, that is mainly, and you can check the different van der Waals surface. Like if I go to structure, surface, add van der Waals, this is giving the van der Waals surface. So it is showing you the van der Waals interaction areas. So there are many ways of visualizing the protein like this. And the best is this is a free software, so you can download it if you want. So anything else you guys want to discuss?
Which another software you actually named about? Which this studio?
Sorry, your voice is breaking. Sorry.
Actually, which another software you named about? I just noted down Visual Studio and another software you have named that we can use it for. Can you please say it again?
With software like wait, I cannot understand. Which software are you talking about?
You just mentioned little before. I'm just want to know.
I mentioned, I think, Discovery Studio only. Someone else mentioned PyMol. So I'm not sure. Like I was only talking about this one software till now.
You actually, yes, you are saying and you are also demonstrating the usability of this.
Yeah, Disco and PDB database I was showing.
Okay.
If anyone else can remember what he's talking about,
Actually, I think he's talking about the PDB only.
Oh,
because
okay,
you
okay.
mentioned
This
only.
are you talking about this PDB database? Hello?
Yes, yes. Okay,
This
yes.
is not a software actually. You just have to, in Google, you can just type the PDB formula and you will get the PDB database. So, and if you don't know the PDB formula also, like suppose some other protein, hemoglobin, just write hemoglobin PDB in Google and you will automatically get the PDB formula with the, so and if you click that link, the protein data bank database will open. So you will be able to see the structure and all kinds of data about it. So this is not a software. You can just type in Google and you will get it. So there are different, there are other softwares that I can show you. Everyone has ChemDraw, right? Hello?
Ma'am, can you please repeat what you have told?
There is ChemDraw. ChemDraw, do you have ChemDraw?
No, ma'am.
So that is also a software basically for drawing chemical structures. I'll show that also. These are basic softwares in any like chemistry or chemical biology lab that you use. So suppose if you go to structure and convert name to structure, if I want a polypeptide, say ALA, Phi, glue, You have to type the 3-letter codes and you will get the structure. This is the 2-dimensional structure. So like Discovery Studio was for the 3-dimensional structures. In ChemDraw, you can see the 2-dimensional representations. This is mainly for like if you are writing a paper, so this is for drawing the structure so that you can represent some reaction, or like in Edman degradation you saw the reaction, so over there the structures are drawn by ChemDraw. But this is not of any like particular use for understanding the protein 3-dimensional structure. Maximum you can see like this molecular weight you can see. So normally what we do when like Edman degradation, when you are sorry, not Edman degradation, mass spectrometry, what you do is that you break the peptide into many different sized peptides, right? So you can get a tripeptide, tetrapeptide, and so you can check the mass what is supposed to come, and you can check the peaks from the mass spectrometry. So when you compare that, you can find out what fragments you have. So ChemDraw gives you the mass, and other than that, you can get, one second, these boiling point, melting point, and all physical properties you can get, pKa, these things. So it is not much use for the 3-dimensional structure at all. This is just a very basic software for the 2-dimensional representations.
Okay, ma'am.
So
Thank you.
Another one, just added one other software about protein.
what was the other
What?
software?
Someone have mentioned.
Someone mentioned PyMol, but I have not used that actually. So I don't know about that. Is it a free software?
Yes, ma'am. You can download it by education purpose only, ma'am. When
Okay,
you will be
fine.
going to the for the downloading option, they will give you for education purpose, and it is for free for education purpose only.
Okay. So we were actually told that this Discovery Studio was more user friendly, so we had downloaded that. But I will check out PyMol then, if that is better. So yeah, I can do that. And
I also don't know like whether it is better than Discovery Studios, but actually we are students, so we generally use the PyMol for visualizing 3D structures.
But this PDB you can open in PyMol.
Yes, ma'am. Yes, a PDB
Okay.
file we can open in PyMol.
Okay. Then probably I'll check it out. But yeah. What other softwares do you use?
Ma'am, actually, currently I am in 6th semester. So in previous semester, we used homology modeling. For homology modeling, cluster W, those things we have used in previous semester, ma'am. For homology, and also I don't remember the name, ma'am, currently, and that is for, ma'am, the GOR method, GOR method. That is also online, completely. That is also user friendly, ma'am.
Okay. So have you done these molecular dynamic simulations and all these things?
No, ma'am. That's why I opted for this course. Only
I
I have visualized only the 3D structures by taking from only NCBI or PDB those files and also UniProt. But I have not studied the things like the analysis and those things. So that's why I opted for this course only, ma'am.
Okay. So if anyone has any doubts about the second week also, like second week was mainly about proteins and all. So if anyone has any doubts about that also, you can ask now. Just clear all your doubts.
Ma'am, can you please tell about the alpha angle, the gamma angle, the psi and phi
The
a little?
dihedral angles, no? Okay.
Yes,
I'll
dihedral
tell
angles.
you. 1 second.
Yes, ma'am.
Okay, so this is showing the peptide bond, which is the peptide plane. And this phi angle is basically from this carbonyl carbon to the next carbonyl carbon. So there are 4 atoms here, and angle between 4 atoms is called a dihedral angle. So if you want to understand the dihedral angle better, I'll show you from Discovery Studio. 1 second.
Okay, ma'am.
I'll show the dihedral angle. If you see it from the 3D thing, it will be easier to understand. 1 second. So from carbonyl to carbonyl, this angle is showing minus 79.46. So this is called a Chi angle, which is between these 4 atoms from carbonyl to the carbonyl. For the this is the phi angle. For the Chi angle, it is from nitrogen to the next nitrogen. Here it's 144.05. And then what are the other angles? Omega. Omega is basically, this is called an alpha carbon, which is the one directly beside the carbonyl. So omega is from one alpha carbon to the next alpha carbon. This omega angle, I'll delete the others. This is the omega angle, and here it is near 0 degree. So this is called so this bond is in the cis conformation. So you learned that the amide bond has partial double bond character. So if this partial double bond is in this conformation, then it is a cis conformation. And I'll rotate this bond. If it is in this conformation, then it is a trans conformation. This is trans conformation. So this is the omega angle. And 1 second. Another angle is the chi. Chi, you will understand. Chi is you start from nitrogen and you go to the side chain. So you know this is the main chain of the protein, which goes from carbonyl, NH, alpha carbon, again carbonyl, but this is the side chain. So here it is leucine, which has a CH2, CH, then 2 CH3 groups. So it's a leucine side chain. So if you start from the amide nitrogen and go to the side chain, this is called your chi-1 angle. It's near minus 60. So these are all dihedral angles because it is basically angle between 4 atoms. So 3 bonds. So to recap, if you start from carbonyl carbon, go to the next carbonyl carbon, that is a phi. If you start from nitrogen, go to the next nitrogen, that is a Chi. And if you start from C alpha and go to the next C alpha, that is omega. And chi is not, here only the backbone is shown. So chi can't be seen. I'll show you chi. Right. Yes. So chi is from nitrogen. If you go to the side chain, then you this picture is not very good. But if you look at it here, this is chi. This angle is chi. So these are the dihedral angles.
Yes, ma'am. Thank you, ma'am.
Mm-hmm. Any other doubts from the proteins class or from week 1 or week 2 or week 0? Did you try solving the week 0 questions?
Yes, ma'am. Solved.
Okay. So no doubts in that question, right?
No, ma'am.
Okay.
Ma'am, can I ask a question?
Uh-huh.
Ma'am, are you going to like repeat these angles in the next lectures also, ma'am, like for the problem solving purposes?
Yes, like for week 2, we are supposed to discuss the week 2, today I discussed week 1 summary and problems.
Yes. Yes.
Again, in week 2, I will do week 2 summary and problems.
Okay. Ma'am, can you please tell about like electrostatic surface potential?
Okay, so I don't have the slide for that. I'll just note it down. I'll make the slide for the next uh, lecture. Okay,
Because
electrostatic.
ma'am, because ma'am, I have opted for the answer, but I don't know much about it. It is the week 0 question.
Okay, okay.
It is in the week 0 question.
It
Yes,
will be
ma'am.
easier for you to understand from the slide. So I'll make the slide in my next lecture, and I will explain it properly at that time.
Okay, ma'am. Okay, thank you.
Sure. Everything else was clear, like Ramachandran plot and all that.
Actually, ma'am, I have not studied the Ramachandran plot very matlab, in a very efficient way.
Okay.
I will, that's why, ma'am, I am not able to ask the questions.
Okay, okay, fine. It's fine.
I have to
Anyway, next week we will discuss all that. So, in the first week, mainly I saw this this year's assignment. I saw that they have asked all these questions, that who discovered that Robert Hooke, like all that history question was there in this year's assignment, right?
Yes, ma'am. Yes.
But I don't think you have to remember year and all that. So actually, this question I was very surprised by, because it is not at all possible to remember this. So you can remember the amino acids, and you can remember the amino. That is important, by the way. You have to remember all the amino acid structures, 3-letter code and 1-letter code. But I don't know how it is possible to remember this. That which is the nucleotide that is coding, because this was the question last year. So I don't know how it is possible, because you
Yes.
guys have an exam also, right?
Yes, ma'am. 30th
Uh,
April.
so in the exam, if a question comes like this, that what codes for proline, there are so many options. So I don't know how that is possible. But this
Ma'am,
amino acid, what you are saying?
ma'am, are the in final exam the questions will be subjective or objective, ma'am? Like,
Like last time it was objective only, but this time you once clarify with sir,
okay,
because
sir.
it can change this time, no? So just clarify that once.
Ma'am, how to clarify that?
You can mail sir. I think sir's email ID would be present in the or you can mail the Lakshmi ma'am also. You can ask, actually. I think better than asking sir, because Lakshmi ma'am is the like coordinator for this NPTEL thing, no? So you
Okay,
have
ma'am.
Lakshmi ma'am's email ID. You can just ask her that once.
Okay, ma'am. Okay.
But this you have to remember, that this amino acid, these questions can come, like it had come last time also, that what is the structure of leucine, and you are given 4 options. Or what is the amino acid that has the single letter code L. So that you have to remember.
Okay, ma'am. Okay.
Hmm.
Ma'am,
And what are
I
you
want to
saying?
ask. I want to ask something, ma'am. Actually,
Mhm.
ma'am, I am a biotechnology student and
Okay,
currently in third year, ma'am. But
third.
ma'am, I am very much interested in going for higher studies, like for the structural visualization of these things, like proteins, amino acids. Ma'am, are there any option? Because currently I am in third year. Ma'am, I have to go for either GATE or for masters, and after that or before that we have to go for the CSI and NET and those things. Ma'am, are there any process that I can be easily available, like I can be easily able to visualize these things or work in some labs so that I can get benefited to know the things?
Like internship is an option for you in third year?
Yes, my internship is an option, but it is very tough actually to get internship in prestigious universities like ISAR or IITs.
Okay.
So
See, I don't think I am an integrated PhD student. Okay, so I am an integrated PhD student in IISc. So before coming to IISc, I did not know all this visualization software, but I came here and learned. So that was not a problem. So you can, like in your MSc, you will learn. You are in BSc third year, right?
BTech.
BTech,
Actually.
sorry, BTech third year. So in MTech, uh, like I also came without any knowledge up to my third year, but then I joined this integrated program and I was taught everything. So I think yeah, that won't be a problem. Otherwise, tutorials are available. Like the that is the, I normally do molecular dynamics simulation using a software called GROMACS. So that is also a free software. So you can find tutorials about all this on YouTube. So most of the things I learned from YouTube or tutorials only. So you can try that also. It is not that difficult.
Okay, okay.
So you can mail me. My email ID is, wait, you have my email ID? I'll
No,
you
ma'am.
you can mail me. I can give you some suggestions. I'll just write my email ID. With doubts also, you guys can mail me. So I'll write my email ID. This is my email ID. So with your doubts or anything, if you want to ask, you can ask at this ID.
Okay, ma'am. Thank you, ma'am.
Okay, okay. If any like you guys are having any problem solving the assignment questions, current assignment questions also, if you want, you can ask. Like if there is some problem solving. But I think as soon as you solve, they give you the answers, right? Like they will tell
No,
you this is correct, this is not correct. They will tell you like that.
no, ma'am. Actually, it goes for the review purposes, and then the result comes.
Okay, I
Till now,
thought because assignment 0 was designed like that, that you solve and it immediately tells you if the answer is correct or incorrect.
yes, ma'am. For it was for
So,
assignment 0, but
okay, okay.
not in other assignments, ma'am.
Hmm. So,
Ma'am, is there the availability of the uh, previous set of questions is there in site? Can we get that?
previous years, I'll ask once. Like I have it, but I don't know if I'm allowed to share it. So I have to ask Lakshmi ma'am once. So you can just mail and ask me, and once I confirm, I will let you know.
Definitely. Thank you.
So these slides that I have prepared, they will all be available to you via Google Drive. So the slides and the lectures, if you need, they will be available. So I will share the link. I think we have been given a Google form in which to fill the details of these slides and the lectures. So you can access that anytime. Because
Please give
I've
the
been, sorry.
Please give the link for that.
Yeah, I have to anyway give the link to the NPTEL committee, and I think they will share it with you because I know some of the slides I went through a little bit fast. So if you want to go through the slides later slowly, you can go through that.
Even ma'am, in previous session, which was actually in last 3 days before, in that day also was my network problem was there, so that's why I could not get that link even. So if it is possible to get all the links, then it will be helpful if we want
Yeah.
to get the slides.
I think Lakshmi ma'am will share that, because we have been told to make these links available to everyone. So we will submit it to the NPTEL committee and they will, I think, share it with all of you, because they have asked us to do that.
Actually,
And
the um, this online meeting links are provided, but for
Mhm.
notification come in timely. But actually, there is no other mail they have been sent to us. So if it is possible that we will get every document and your lectures and also the slides, it will be better that
Uh-huh.
it will be additional knowledge sharing
Yeah,
tool
yeah,
for us.
definitely. I will inform the Lakshmi ma'am. I'll tell her. Okay.
Lastly, I suggest them if there will be a single drive or a single group of mails can be created by our all the students who are enrolled in this program. If it will be
Mhm.
available one by a mail also, then it will be easier
Ah,
for us. It will be jotted down and it will be
yeah,
some
yeah.
marked in a way that it will be available.
Okay, okay. I will talk with her. I'll talk with her. Okay. Mhm. And you know that like there are 4 TS sessions, but in all the sessions, the same thing, more or less the same thing only is taught. So you just attend the one that is most suited for your timings.
Actually, by mail, all the slots are actually given by timing only. So we don't know which slot is actually marked with whom.
Sorry.
Actually, through mail, all the session and its timing only they have disclosed. So we don't know which session is actually tagged with which part. So we are attending maximum of the session. But
That is what I'm saying. You don't have to do that. Like, I think there is a session on Friday, Saturday, Monday, and Tuesday. So you just attend the day that is most suitable for you. Because in the TS sessions, we have been told, all of us have been told the same thing, that discuss a summary of the lecture and then discuss practice problems. So
that's
today
okay.
I
But
saw that. Hmm.
slide and the first slide was even the second slide was little bit different because everyone has their own good knowledge which you all are sharing.
Okay, okay,
And I
okay.
thank
Fine.
you
Yeah.
and
Yeah, if you have the time, definitely you can attend. But yeah,
yes,
whatever
that
is
problem,
easy.
what you were saying, it is also a problem for
Huh.
everyone. I because everyone has the different agenda and educational purpose.
Hmm. Exactly.
But it is also a good thing to attend everything because maybe there is a similar or Mars
Hmm.
something, Mars topic. But it
Hmm.
is good that everyone has some added knowledge also in the
Hmm.
different.
Right. Like basically today I saw that I had prepared only 10 questions. So 10 questions got finished very early. So next class I can prepare more questions. So this is the first class for me also. So that I understood. So next class maybe like 25, 30 questions I will prepare. So that because the practice sessions are a practice for you guys also. So yeah.
Thank you for.
Mhm. Yeah. So I have to be online till 8. So if you want, you can ask anything. I don't know, any questions.
Ma'am, can you give one idea that if there is any nomenclature module for genetic recognition like DNA, like RNA, the group of DNA, group of RNA, and also the for protein, like our biological tree for bacteria, viruses, funguses have. Like that, is there any nomenclature module for this molecule?
No, like that. NPT in NPTL, as far as I know, there is nothing like that. Nomenclature is taught alongside normal class only, but that is very basic nomenclature. Like sir had discussed, like trios, pentos, hexos. It is like that. But you are talking about more basic things, right?
Yes, actually, the total cross section. Suppose I am telling about all the discovery of the protein and its name available in throughout the world's knowledge. Is there any database where this nomenclature or this biological tree, like nomenclature tree or groups of molecular grouping, is present for the DNA for RNA?
No, no. As far as I know, there is nothing like that. Like everything we learn as we are learning only. When you were learning carbohydrates, you learned some of the molecules, but like that, general database for everything, there is no such module.
Yes, I actually want to learning about more about if there is any in-depth grouping about the molecules which is discovered molecules actually or the present molecules which we know till date. So is there anything or can you please talk to the team if there will be any program that it will can be
Okay,
constructed
okay, okay.
to know about this constructible knowledge that will be helpful for all of us, all the scholars and everyone.
Okay, yeah, that is a good suggestion. I will, yeah, because there are, this is a 12 class module. There are 4 class modules also. So it can be a short module like that. So yeah, I can try, I will mail Lakshmi ma'am.
Actually, this is, I think, throughout my working knowledge, I got to understood that this is actually the need of the future because if this will be constructed, then it need to be available for all the researchers who are working with this, but because this will be helpful. Because
Hmm.
if one want to group it or know it about or want to denote some particular molecule or particular protein or whatever the molecule is present to clarify or compare comparison with that the developed molecule or enumerated out molecule from the result part, it will
Hmm.
be better that it can be a matching version that will be work as a ladder as well as a standard.
Hmm. Okay. Okay. I'll talk to Lakshmi ma'am about this. Yeah.
Can you ma'am please share the Lakshmi ma'am's ID also? Actually, I don't have.
Just wait a minute. Okay. I'll just search for it. Yeah, this is it. Yeah. So yeah, I think there are no more doubts for anyone. So anyway, if you have any doubts in between, you can send me your questions in my email ID, and in the next TA session, I will discuss those doubts. So I think
Okay.
that's okay. That's it for
But
today. Yes, you were saying something. Hello?
Say it again. ID, Lakshmi and site.
Okay. Yeah, wait. Sorry. Yeah, just note it down.
Okay, thank you.
Okay, sure. Okay, see you in the next session.
Thank you, ma'am.